So it's important to do some filtering, and I will come to the filtering for the mitochondrial gene after filtering for the expression or some of the standards. And the way that we do it is by specifying a cutoff for a minimum number of genes using filter cells function in the preprocessing module, and then we specify the AND data and also minimum number of genes, which is 200. It's not like written on a stone, you should use this one. It is just a standard. Depending on your data set, you can play around with these values. In addition to that, we have filter genes, which will require a gene to be expressed in at least 3 genes. So there we go. Now we have filtered out 1300 cells and 14,000 genes. Remember that we are working on a small subset of the data, so these values, if you run the whole data set, will differ. So the next thing which is important to do is to see the highly variable genes, because we are going to use this information in downstream data analysis. So it's as simple as using this function, highest expressed genes, and then we specify to annotate top 20. So or to visualize. So what we see here is the box plot showing the percentage of total counts. As you can see, we have mitochondrial genes and MALAT1. So what's the story about MALAT1? MALAT1 is a nuclear link RNA, and its high expression may not be surprising because it is frequently detected in poly-A captured single-cell RNA-seq data, and it is the case in this data set since we are using 10x genomics data, and it is poly-A captured. So we need to filter this out, but before specifying given genes, we can apply a cutoff for the mitochondrial genes. So in this case, we say 20%, and also we apply another cutoff for the ribosomal genes, which we require them to be above 5. So and then we can see how will the numbers look like after this filtering. So for some of you who may not be familiar with Python, that's the way that we do it in the filtering. So we say, okay, from the object and data, which when we are passing it like this from observed data, I have access to the indices across the data frames or the slots that we have. So this is on data, and when we are using the indices, we get access to the rows or columns. So here what we are trying to do is that we say, okay, at the column in observed slot, check the percentage counts mitochondrial column and keep those that have a value less than 20, and the same for the counts ribo for the values that are above 5. And we do it in step by step. So after this, instead of 12,000 cells, we have now 7,431 cells. Now let's look at the distribution of these QC metrics after filtering. There we go. We don't see super big difference in terms of distribution for genes by counts in samples 15 and 16, and also for the mitochondria we do not see this long tail of the distribution. Again, for COVID-19 we see that there's a tendency for lower percentage of mitochondrial genes. So maybe here we can take a break. I suggest that you take a break as well. Drink a tea or whatever you like, and then we will be back in a few seconds. It is shortest break ever, and it is not real because I trimmed the video. So we should make a bit longer break. So anyway, I'm not here to talk about the breaks, but now let's continue and do some additional filtering. So we talked about MALAT1, and here we are going to exclude that. And the way that we do it, we identify the indices of the rows, and that's in the var name, sorry, it should be columns, which starts with MALAT1. we save this, and then for mitochondria we do the same, and we require from this we by this approach to capture or to fetch the IDs of the columns or genes that start with MT, and the same for the hemoglobin genes. But here we are not specifying whether the gene starts with hemoglobin, but as long as it contains HP, then we store the data here in this variable HP genes. And then we do the removing, and then here we have an NP, which is NumPy. I can show it to you where we imported, just for the record. NumPy, we saved it as, or we imported as NP. And then, or maybe I can navigate through this outline. Okay, we go to filtering, we filter the genes. Yeah, there we go. Okay, so then we save it as indices in the remove, and then we just concatenate, and then at the end we do the invert. So we say, okay, whatever indices you have in the remove, invert it, and those will become the indices or rows or columns that we are going to keep. This is column because we are using the gene names. We haven't been filtering the cells in this chunk. So there we go. From 33,000 genes or columns, we ended up with 19,000, which is a bit more realistic since we are now targeting the proto-encoding genes. Now let's save the data. The way that you do it is to just use the write method and from the data, and then here we are specifying the path, os.path, join, and the rest. So this will save our data, and next step is to check the sex of the individuals. It is important, sometimes you need to do it to match the metadata with the sample, and in case there is a swap or some handling, some technical problems, and you can figure this out at this stage. In addition to that, you can also, if you do not have that metadata, then you can deduce the sex and then use it for downstream analysis in case you want to monitor the differences at the sex level. And so we use Biomart, and then we specify the species and different type of data that we would like to extract from the gene ID to chromosome, and then we set the index to be external gene names. How it looks like this. So we have the external gene name as the index, ensemble gene ID, position, and so on. So here we have 86,000, including all possible annotated genes. So with this information, now we can intersect the information that we have from the Biomart with the gene ID that we have in our data object. And then from there, we add a new column to the observation slot and calculate the percentage, as you see here. Moreover, we extract the information of ZIST. What is ZIST? I think I went through this very quickly. ZIST is inactive specific transcript, which is expressed on X chromosome, and this can give us a proxy to the sex. So here we do the same. We just check whether there's an expression of this gene in given samples or not, and then we plot it. So that's, again, we are plotting using a scatter. We have x-axis the ZIST count, and then on the y-axis we have percentage of Y chromosome. So for samples that we tend to have ZIST count, we expect to see not much Y expression, chromosome Y expression. But to be honest, I don't like this type of plotting. I prefer the violin plot, and here we pass ZIST and percentage of chromosome Y, and we group them by sample, and I like this. It's a bit more a bit easier to read out. So from this we can see that chromosome 1 and 17, it's a bit shifted, and then 19. Let's change that to 90 degrees, it will be easier. Okay, there we go. Yes, so these 3 samples have no expression of this, but you do not see the same picture being reflected when we are looking to chromosome Y. And why? Because when we are extracting this, we also have pseudo-autosomal genes, so it does not give us a clear picture. But for now, we go with the ZIST picture that we have, and we label these samples 1, 17, and 19 as female.